Biology, Part 234. The development of rapid techniques for analyzing the amino acid and sequences of proteins gave a new opportunity for investigating evolution. Well, the figure shows a scheme of describing the probable history of the hemoglobin molecules of several vertebrate species. This is produced by a computer analysis of the sequence information, and the numbers on the number on the branches of the tree are minimum number of the amino acid substitutions required to produce a new molecule from the one at the previous branching point. Each substitution being the result of a single gene mutation, there is a change affecting just one base pair in the DNA sequence coding for the protein. The important point is that the relationship revealed are precisely those expected from the taxonomic and fossil evidence. Studies of the other proteins such as cycloromes have given essentially the same results with other organisms. Comparative anatomy. It is a feature of evolution that the same structure is often adapted and developed for a number of distinct functions. The basic component of the vertebrate skeleton are common to the thousands of different species, but the sizes and the shapes of the individual bones can vary enormously. For instance, some of the visceral arches supporting the gills of the primitive fish act as jaws in the bony fish, or as ears in the ossicles in the mammals. Others survive as the cartilage support for the tongue and the larynx. So this is how we show the various specialization of the vertebrate limbs. The first plan is that then we have got the embryology. The embryonic development of the vertebrates is extremely uniform. For example, all early embryos have branchial grooves and internal gills, pouches. In fish, these meet to form gill slits and later become an important part of the respiratory system. On the other hand, in terrestrial vertebrates, the grooves and the pouches almost disappear, so that the only traces which remain in the adult mammal are the eustachian tube and the auditory canal. Human embryos also pass through the stage in which they possess a two-chambered heart and a tail. The differences between the vertebrate species are best interpreted as the variations on a theme. Certain mechanisms are inherited from a common ancestor. These can be modified but not scrapped completely. Well, there are vestigial organs. Vestigial organs are present both in the embryo and in the adult organisms. They have no known function, although they seem to have had a function in the past. The coccyx of the human skeleton is useless tail remnant, while the appendix, which is corresponding to the caesiums of the herbivores of the animals, is too small to have any useful role in digestion. The vestigial legs bones are found in snakes but make no contribution to the locomotion, which depends exclusively on the movements of the rib cage. And it has got its link with the previous species. Ostrich wings and the partial development of an egg tooth in the embryos of the marsupial mammals are additional examples. Next we talk about the biogeographical evidence. Biogeographical evidence. The distribution of the living organisms has already been referred to our tutorial as important supporting evidence for the evolution. Briefly, the land masses of the earth can be divided into several main regions, each with its own characteristics of life forms. The differences between the regions cannot be explained by climate alone, but are the result of the continental drift, which isolated primitive ancestral populations.